Good morning. Welcome to the Baker Institute, and thank you for joining us so early in the morning. The Baker Institute has had a long working relationship with the Texas Heart Institute. One of the first science and technology conferences featured work from the late Dr. James Willerson, and it was using stem cells for cardiac health. So today's event is the 8th in this series. It's focused on regenerative medicine and policy. This is a long-term collaboration between the Baker Institute and the Texas Heart Institute beginning back in 2016. So the series, while very productive and educational, would not have come into fruition without funding and support from the Hammond Foundation, which we're very grateful for. The collaboration has resulted in not only talks like this, but a number of peer-reviewed publications and the dissemination of these issues and research, not just here at the Baker Institute, but around the world. So before moving on, I'd like to thank the organizers for today's event. This includes Carrie Sprung from the Texas Heart Institute and Kirstin Matthews, who's going to be one of our panelists today, who organized the panel discussion. And I'd also like to thank the Baker staff who make these events run so smoothly. These individuals include Jordan Traylor, Caitlin May, and Serena Storm. Moving on, moderating today's event and to introduce our panelists is Maya Pomroy. Ms. Pomroy is a graduate of Rice's MBA program and an award-winning host and journalist. She founded The Public Lead, a library of community stories, and she is also one of the hosts of Rice Business's podcast, I'll Have You Know. With that.
Thank you, Dr. Ho. Welcome, everyone. We are delighted to have you joining us for the 8th annual, as Dr. Ho has said, Stem Cell Policy Series, which, as she mentioned, is a collaboration between the Texas Heart Institute and the Baker Institute for Public Policy. This has been sponsored by the George and Mary Josephine Hammond Foundation, as mentioned. Our topic of focus today is stem cell therapy for cardiac care. Our discussion will be led by experts in the field of cardiology, stem cells, and the interrelatedness of public policy and its implications. We are joined by physician scientists Dr. Deepak Srivastava, Dr. Emerson Perin, and Baker Institute fellow and policy expert Dr. Kirstin Matthews. We will begin with Dr. Deepak Srivastava, who will share advances in stem cell cardiac research, followed by Dr. Emerson Perin, highlighting advances in regenerative medicine clinical research for cardiac care. And then we will close with Dr. Kirstin Matthews, who focuses her research on work on the ethical and policy changes in the space. She will focus on the discussion of reflections of the past and a review of current issues. We will have a question and answer time at the end of our series, so anyone that's watching online can submit their questions along with our audience here today. So let's begin. Our first speaker is Dr. Deepak Srivastava, a non-resident scholar for biomedical research policy at the Baker Institute, president of the Gladstone Institutes. He's also a senior investigator and director of Gladstone's Roddenberry Stem Cell Center. He's a professor of pediatrics and of biochemistry and biophysics at the University of California, San Francisco, and holds the Robert W. and Linda Maley Distinguished Professorship. Dr. Srivastava's lab used genetics to demonstrate a decrease in dosage of some cardiac development regulators, causing cardiac septal defects and valve disease, and is now using pluripotent stem cells to discover the mechanisms of disease. His team reprogrammed non-muscle cells in a mouse heart to function like heart muscle cells, effectively regenerating the heart muscle after damage, which is fascinating. Additionally, he co-founded a biotechnology company to help find new cures for human diseases. Before joining Gladstone, he was a professor in the Department of Pediatrics and Molecular Biology at the University of Texas Southwestern Medical Center in Dallas. He has been named an endowed chair at both UTSW and at UC San Francisco, and has been elected to the American Society for Clinical Investigation and the American Academy of Arts and Sciences, along with the American Association for the Advancement of Science. Dr. Srivastava completed his undergraduate degree right here at Rice University, go Owls, medical training at UTMB in Galveston, and his residency in the Department of Pediatrics at UC San Francisco. He also completed a fellowship in pediatric cardiology at the Children's Hospital at Harvard Medical School and a postdoctoral fellowship at MD Anderson Cancer Center. Dr. Srivastava will present first on advances in stem cell cardiac research.
Thank you so much. It's wonderful to be back here on campus. It's hard to believe, come on in you guys. It's hard to believe that I was here as an undergraduate 40 years ago. And I noticed that the trees have gotten a lot bigger and I've gotten a little bit grayer. But the Baker Policy Institute is dear to my heart. I've been happy to be involved with it for many years. And the subject matter today that we'll talk about, of course, is dear to my heart as well. And so let me just frame what we'll talk about today in context of where we are more broadly in science and medicine right now. We sit at a moment in time where we finally have the ability to think about curing diseases in ways that we could not before. And specifically for the heart, when people have had heart attacks, we just had to accept that they've lost part of their muscle, and we do the best we can with what they've got left. And that's what's changing now, and that we'll talk about today, is how can we think about actually regenerating damaged hearts and not accepting that those muscle cells are just gone forever, but we can maybe introduce new ones somehow. And so this is the problem that we all face. If any one of us here in the room suffers from a heart attack where a coronary artery gets occluded and all the downstream muscle that's supplied by that vessel doesn't get blood, that muscle dies. And when it dies, it gets replaced by scar tissue, like what you see here. If a coronary artery got occluded here, then you'd have the red cells, which is muscle, would die, and it'd be replaced by these purple cells, which is scar tissue, essentially. And that's what we deal with. And so you can imagine then the heart can't squeeze as well. People, if they survive that, they develop what we call heart failure, where the heart just can't pump with enough force, and that leads to a slow but steady decline, ultimately with a heart transplant being the ultimate treatment for this. But there are very limited numbers done in the United States every year, about 3,000 heart transplants in the whole country. And many countries around the world don't even offer that for economic reasons and sometimes for ethical reasons. And so the question is, can we somehow not accept this situation and rather have new muscle cells be created in the heart that would then replace this scar tissue? And in the last several years, there have been several different approaches that scientists across the world have taken. And I'm going to touch on a couple of those today, summarized here. One is that if you could take stem cells in a dish and turn them into beating heart cells, you might be able to introduce those into the heart and just create new muscle that way. And we'll talk a bit about that. There are two other approaches that I'll touch on that rely on the cells that are left over in the heart that are still there, that are already in your organ. Either the muscle cells that are present, and one approach is to get them to create more of themselves, right where they are, or to take cells that aren't muscle, or never going to be muscle, and get them to be what we want them to be, and turn them into new muscle right where they are. And I'll briefly touch on those three approaches. And later in this session, you'll hear from Dr. Perin of a different approach where you're trying to modulate what's left in the heart and improve the function using stem cells as well. And so let me talk about the first, which is what we call pluripotent stem cells. And these are what many of you might be familiar with, come in two flavors. One is the embryonic stem cells, which is cells that we might harvest from a 5-day-old human embryo and take that human embryo in a dish and isolate the cells. And those cells have the property that they can turn into every cell type in your body, including heart cells. You guys might remember some years ago there was a lot of ethical concern around those type of human embryo derived stem cells because you were destroying a 5-day-old human embryo. And depending on when you believe conception starts, and we'll talk about this a bit later in Kirstin's talk, that could be objectionable. And so one of our investigators at the Gladstone Institutes where I am, Shinya Yamanaka, made a really seminal discovery some years ago where we found that we could take a teaspoon of any one of your blood, as blood sample, or a sample of your skin, and in a dish introduce just 4 proteins into those cells. And those proteins had the magical ability to take those cells back in time. So they were now reset to be almost exactly like your own 5-day-old embryonic stem cells. So it obviated the whole ethical debate in part around using human embryonic stem cells, and it gave access to people's own, your own stem cells, even though you're now an adult. And so this revolutionized the field of stem cell biology worldwide. He received the Nobel Prize for this discovery in 2012, and it's allowed us now to take cells from any individual with the disease and make their own embryonic stem cell equivalents that then could be turned into whatever cell type we want that to replace what they might be suffering from. So we've used these in 2 distinct ways. One is, you can imagine if somebody has a disease, say they have Alzheimer's disease. Ideally, you'd like to get their brain cells and figure out why do their brain cells die and what drug might be able to stop that. But obviously you can't take a biopsy of somebody's brain and do that. But now we could take the cells from somebody with Alzheimer's, make their stem cells in a dish, turn them into the exact type of brain cell that is damaged in Alzheimer's. We do the same thing for heart disease. And now in the dish in front of us, figure out why it happened and test drugs on it. It's the ultimate in personalized medicine. The other way we've used these is to turn these cells into whatever we want, heart cells if they have heart failure, insulin-producing cells if they have diabetes, brain cells if they have Parkinson's, and use those to transplant back into the patient the cells that they've lost. And we now are able to quite efficiently take these cells and turn them into basically any cell type we want in the human body with pretty good efficiency. And I'll give you an example of us turning these cells into beating heart cells in the dish. And that's shown here. What you're seeing here are thousands of cells, actually, and each one is very small. They're connected to each other, and I'm going to play a movie for you in a second that'll show you that these are actually beating heart cells in the dish, and these used to be on somebody's skin. And I might need somebody's help to make this play. Yes, thank you. So here you can see these are cells that are beating together with its own heartbeat that's similar to ours. They're electrically connected with one another, and just like our hearts, they beat in unison repeatedly. These used to be on somebody's skin. This is like science fiction, but it's something we can do, and we can make billions of these cells now. And so you can imagine now taking these cells and transplanting them back into a damaged heart and replacing the lost cells. And so there are currently trials going on in humans now for these kind of cells, both for heart but for many other cells all over the world for lots of different diseases. And they're just reading out now, so the next 2 or 3 years, you'll see readouts from these clinical trials, and some will work and some won't work, I'm certain. There are about 3 or 4 trials going on worldwide using the kind of cells I described there, transplanting them into damaged hearts in people. And we'll see what those shows. So far, I'm hopeful, but there are a couple of concerns with those. One is that the cells that we make in the dish are not really fully mature like our own adult cells. And so they don't beat with the strength that we like, and they don't electrically connect so well with the cells that already exist within our heart. And so in animal studies, they've caused some rhythm problems. And so right now they're going into people who already have a defibrillator in place. In case that happens, they can get shocked out of it, et cetera. So there are some safety issues, and efficacy issues, but we'll see. So that's all happening. My own laboratory decided, as we observed this, to take a couple of different approaches, as I alluded to earlier, where we're trying to harness the body's own cells that are already in the heart so that we're not actually putting new cells in, and that might overcome some of those connectivity issues that I mentioned. And one of the ways, well, here are the two different ways we've taken. One is to say, as I mentioned earlier, let's take the muscle cells that are already there in the body, in the heart, and just get them to divide again. And one of the things that we know happens is right after birth, the heart muscle cells stop dividing and never divide really ever again substantially. And that's why when heart muscle dies, you can't get more of it. And so the question we ask, could we just alter that and get the ones that are there to divide again like they used to do when they were in the embryo? Because at that time they are dividing. And the other approach is to get these cells that might make scar to not make scar, but rather make new muscle. And it turns out that we found some years ago that a combination of 4 different proteins that are essential in the embryo for getting these cells to divide, the heart cells to divide, if we reintroduce those in the adult heart, we could actually get adult muscle cells to efficiently divide again, which was really quite amazing. And so this worked really well in animals, but we used a virus to deliver these, and it kept making these proteins. And you can imagine that, there's a problem if cells keep dividing, right? What's that called? Cancer. Okay, so that was really never a viable approach in humans. And so we were stuck then. Well, fast forward a couple of years, COVID happened. And what happened in COVID is we got these new types of vaccines using what we call mRNA, and now everybody knows what mRNA is, but it's basically a temporary code for a protein that you put in the body, and 3 days later it's not to be found, it's gone. And during COVID, the companies found that you could package that in what they called a lipid nanoparticle, which is basically a casing that allows it to enter our cells efficiently. Those technologies didn't really fully exist in clinical settings before, but after COVID they did. So now we've revisited this problem with that kind of delivery because it's perfect for this. The proteins are there for a few days. We can use a lipid nanoparticle to deliver it, and then it's gone. So you don't have the cancer risk. Well, the problem was there wasn't a lipid nanoparticle, we abbreviate that as an LNP, that efficiently got into the heart. And so we went on a campaign a few years ago to find a new LNP that would do that, and we found one. And what I'm showing you here is a heart that you'll see in a movie in a second, where we've delivered something that's red, shows up red, into a heart by injecting it in a vein. And this product now, LNP, hones into the heart in the area of damage. So in this animal heart, we've closed off the coronary artery for a while, about an hour, and then opened it up again, much like if somebody had a heart attack and they got a stent, like Dr. Perin puts in to people. And then we've asked, where does this thing go? And so here's the movie, and the heart is in this whole area. The bottom part here is where the infarct would have occurred, the damage, the heart attack, and you can see there's a lot of red in there, and the red in big cells, which are the muscle cells. There's also red in the other cells, the scar-forming cells, so we could use this to deliver to a number of different cell types in the heart. And we're zooming into that here, and you can really see it gets in there well. And so now we're delivering these factors that can induce the muscle cells to divide again with this strategy. And so it's worked well in small animals, and now we're advancing it for bigger animals. So that's the second way we could approach this. The third way we can approach this is by getting the scar-forming cells that are already in the heart to turn into new muscle right where they are. And we call this reprogramming, where we're basically reprogramming cells to be something that they're not usually. And the reason that we are attracted to this is that it turns out that each one of your hearts is made up of muscle cells, as you can imagine, but only half the cells in your heart is made up of muscle. The other half of all the cells in your heart are what we call fibroblasts, and these are the cells that form the architecture of the heart, and they send important signals to your muscle, but they're also the ones that get activated after injury and go and make the scar that we talked about earlier. And there's so many right there where you want them. And so the question we asked is, could we find a way to get these cells to not be fibroblasts or scar cells, but rather switch their fate? So now they become beating heart cells right where they are. And then you could sort of regenerate the heart from within. And in fact, it turned out to be pretty simple. A combination of now 3 different proteins that the body normally, the embryo normally uses in the fetus to make a heart cell. If we put those back into an adult non-heart cell, it converts those into a new beating cell that looks like a cardiac muscle. And so now we've done this in mice, in rats, and in pigs, and it's worked pretty well. And so we've put this into a biotech company in San Francisco that we called Tenaya Therapeutics, and they are taking this forward into a clinical trial. And one of the things we found in our studies is that unlike the stem cells that we introduced, these cells, maybe because they got created from within, electrically connected with their neighbors, so they beat in synchrony, and they get more mature than the stem cells that we're putting in. So we're hopeful. Dr. Perin has been collaborating with Tenaya actually to test this in pigs, and we're working on how best to deliver this efficiently into the heart with the catheter from inside the heart, much like we would do when we're putting a stent in. And so those are 3 of the approaches that I wanted to mention to you, all different, but I think it's important to attack this problem from multiple angles because this is a difficult problem and we're not sure which one is going to work best, and it's best for us to come at it from jointly. And I'm going to leave you with one thought that maybe we can come back to in the moderated discussion period, and that is, as we develop these, what I'll call curative therapies, where we no longer accept that you've lost muscle and now you have new muscle, these therapies are going to be delivered one time, not a pill you take every day. And so with that, and they're expensive to make, and with that comes the issue that they're going to be expensive to take because they cost a lot to make, and a company that's invested billions of dollars has one chance to recoup their costs because you're not going to come back to them if this works. Right? And so inherently these are going to be very expensive. And so what worries me the most is that we'll figure out the science of how to do this and cure people's diseases. And then if there's not innovation on the other side of delivery and finance, then people aren't going to actually get these drugs because insurance companies or a government won't actually pay for them. And even if they pay for them here in this country, there are people on all corners of the world that aren't going to get these medicines if they cost $1 million. And so this is something that as a field, I think we need to solve. And brilliant minds like Vivian Ho and others here are hopefully going to help us figure that out. So I hope that this gives you some insight on where we are today in science and medicine related to cardiac regeneration. And you'll hear more from Dr. Perin about yet other approaches that are being taken. Thank you very much.
Thank you so much, Dr. Srivastava. You said it was science fiction, but now it's going to be science nonfiction. That's all of our hopes. So our next speaker, Dr. Emerson Perin, has provided leadership at the Texas Heart Institute for more than 25 years, and as an alumnus of the Texas Heart Institute's Cardiovascular Disease and Interventional Cardiology Fellowship programs. He's a world-renowned cardiologist known for his leadership in the development of stem cell therapies for cardiovascular diseases. He currently serves as the director of the Center for Clinical Research, director of the Stem Cell Center, and medical director of THI appointed by the Board of Trustees. As a pioneer in clinical regenerative medicine, Dr. Perin performed the first stem cell injection in a patient with heart failure in 2001 and subsequently published this first clinical trial to evaluate stem cell therapy in heart failure patients. He has led multiple clinical trials of stem cell therapy, focusing on the treatment of heart failure, acute myocardial infarction, refractory angina, and peripheral arterial disease. Under his leadership, THI has become recognized as a worldwide leader in clinical research in stem cell therapy. Moreover, Dr. Perin was a founding member and principal investigator in the NHLBI Cardiovascular Stem Cell Therapy Research Network, a consortium of 7 sites funded by the National Heart, Blood, and Lung Institute, and charged with advancing the use of cell therapy for treating heart disease. In 2013, he was honored for his pioneering clinical research and named a McNair Scholar at THI and appointed clinical professor of medicine at the Baylor College of Medicine. Dr. Perin will now present Advances in Regenerative Medicine Clinical Research for Cardiac Care. Dr. Perin.
Thank you. That's a hell of an introduction. I'm really not that great. So, first of all, I want to thank Kirstin and the Baker Institute for having us again. And I'm on a mission. It's been a long mission. She mentioned it was 2001 when we did the first patient, and that mission is to cure heart failure with cells. And I'll show you in a video why what got me started on that mission. But let me just give you a little timeline. There's this company in Wall Street called Gartner, and they evaluate new technologies. And pretty much every new technology goes through cycles. When you invent something brand new, it's like it goes first from, you discover something, and then it becomes like the greatest thing since sliced bread. Everybody's interested. There's all this stuff. And so we get this sort of peak of inflated expectations. But then, these things many new technologies are complicated. They're not easily placed into action and used commonly, and so there needs to be a lot of groundwork to get them integrated into what happens. And so you have this sort of trough of disillusionment. Everybody goes like, yeah, what happened to stem cell therapy? I thought you guys had this thing figured out. And then over time, things gradually, we get data, we do studies, and again, this applies for everything, but if we look at cell therapy for heart failure, 2001, we did the first patient in Brazil. And in 2003, we maybe overhyped it a little bit. I guess the media is not good in a sense because it really likes to overhype things. So we were very excited about it. But then around 2010, nothing's happening. We're doing all these trials and we're just figuring out a lot of stuff. Cells are complicated. It's not a medicine. A pill is a chemical you take, and it's a little chemical structure. And a cell is something that has, there's a living being that interacts with an environment and puts out multiple medicines, if you will. So it's complicated to understand that kind of treatment. And so over the years, I've kind of put some dates there where I think we are. And I think we're really getting somewhere. And what I want to show you is, for the first time, you're going to see some unpublished data, stuff that's been accepted to the European Journal of Heart Failure. I'm going to show you some mortality data. The rubber hits the road. I can show you the heart pumping better and this and that and the other. But if I show you people dying less, now we're getting somewhere, and the FDA is taking it seriously, and we're looking at getting things approved. So let's see if this is going to run. The guys back there said... they're magically doing this. This is in Copacabana. And here we go. And these 2 guys are the first guys in the world that got stem cells injected into their hearts. These guys couldn't cross a room, okay? And here they are. This is a Discovery Channel special back in 2000 and whatever, 2 or something. And here they are jogging on the beach. Okay, now, not everybody in this first trial we did responded like this. But that's all I needed to see. So, like I said, I'm on a mission because I know this works. But there's a lot of figuring out to do. Now, so this is kind of represents let me go back one. That represents the hype. But then You know you're not on the right track if you don't have a bunch of people criticizing what you're doing. Here's something that came out. I talked about that trough of disillusionment. This is an editorial in Nature Biotechnology basically telling us, clinical scientists, that we're idiots. We should stop all these clinical trials. We shouldn't be doing this, and this is a waste of time. So that's, we responded with a, and there wasn't an author on this. They just published this anonymously. Anyway, it kind of pisses me off, but anyways, it's life. Okay, so this is complicated, but I just want to kind of take you through. Every box here is a clinical trial that Texas Heart has led. Okay, so these are we've participated in other trials, but these are kind of our trials. And so it all started, how can I highlight things here? There we go. It all started here. This is the Brazilian trial. And then this then we talked to the FDA in 2003, it got published, and that led to a trial here in the US at 2 sites that led to a trial with the NIH network. Now this was bone marrow mononuclear cells, kind of a weak product. Every line here is kind of represents a kind of a different cell that we've used. And so you can see these are fat-derived cells that actually, this is led to approval in Spain for ACL repairs, believe it or not, because these fat-derived cells are very healing. All the soccer players from the Premier League, when you hear that they get hurt, they go to Barcelona, and Dr. Cugat, who we worked with, is approved by the Spanish government to put stem cells in the ACL repairs. When we were doing the trial, the guys that were doing the MRI said, you got to change the timing because we could see that there's a group of people that are healing a lot faster than the others. Anyway, so some stuff exists, But back to heart failure. So what I'm going to concentrate really is I'm going to show you this line right here. And these are mesenchymal cells. And we're going to talk about regenerative medicine, but these mesenchymal cells work in a little different way, and I'm going to give you some highlights of how that is. And so there was a first in man back in 2007 in Australia. Then we did a dosing trial to figure out what the dose was. And you can see, the years are going by here, And then there's the DREAM-HF trial, which we published last year, which is the phase 3 trial, a very big trial of nearly 600 patients. And now the white ones are trials that are about to start, okay? And so DREAM-2 is about to start. And the FDA said, you do this, we'll approve it. So I'll show you the data that's leading to that. And so basically, this is just really to say that this is really a multi-center. There's 52 sites in the U.S. and Canada, double-blinded, sham-controlled. This is a very well, elegantly controlled, rigorous trial of cell therapy that was sponsored by the company called Mesoblast. I have zero conflict of interest. I'm the science guy trying to get this done. And a colleague in San Diego was a co-PI. And guys at Harvard were they adjudicated every clinical endpoint, and we had a data monitoring committee chaired by Dr. Rouleau at the Montreal Heart Institute, who really looked at making sure we're not killing people and things are going okay. This is a study that lasted 5 years, okay? It was based on a lot of background. These, just so you'll know, these cells are mesenchymal cells. They're from the bone marrow, but you get them in a different way. You basically take the bone, you put a needle in the bone marrow, you take out the bone marrow, and with an antibody, you pick out 1 in a million cells, and then you take that cell, and the property of stem cells is auto-renewal. Okay, if you think about it, it's kind of mind-boggling. It's like eternal life, right? So they renew, and you can have a whole cell bank of these cells. So in this trial, we used, because we didn't want to use just one donor, but we used cells from 3 different donors. Again, the first one was done in 2006, and then we did a phase 2 trial that really looked at dosing of these cells, and we figured out the dose. And here's just showing you from that phase 2 trial. You can see the dose: 150 million cells, 25, 75, and these are clinical events. This is death and heart attacks and strokes. And believe it or not, in that 150 million groups over a period of 3 years, kind of miraculous, but nobody in that high dose group had an event. Whereas the control, the people that just got water, tracked with the other 2 doses. So many times there's a threshold effect. You need to get to a certain level. And so we think that dose of 150 million is where we need to be, and that's what we used in the DREAM trial. Now, this is a very complicated slide. I know most of you are not molecular biologists, and I'm not sure I understand all this stuff. But I just want to make a couple of points. And the speaker before me here, Dr. Srivastava, talked about heart attacks. So ischemia, or injured myocardium, ischemic cardiomyopathy, is the most common form of having heart attacks. Number 1 cause of death in the United States is heart attacks. People are living longer because we're doing better taking care of patients, by the way, with prevention, which is another whole story. But everything starts with injury. And as we learned in COVID, there's innate immunity and there's adaptive immunity. And that innate immunity, this injury provokes a bunch of, think of it as a bunch of chemicals and stuff called cytokines and chemokines, and also some kind of cells. And the adaptive immunity part has to do with a certain cell response. This guy right here, the macrophage, this is the main player. And we can transform some of these macrophages with these cells, because now, after 25 years, kind of start figuring out exactly how this works, which was one of the main criticisms, that we were doing things without knowing. We saw the benefit, we want to do it, but it takes a while to sort of put everything together and understand what's happening. So you're getting all these chemical substances here, and they are working together with some cells that are in the heart, and basically that injury, there's these resident macrophages, and you can see right here, these cells, they're there all the time. Because I'm going to show you the DREAM trial now, and this is a 5-year-long trial. We do 1 injection. The thing lasts for, I'll show you data for 5 years. You'll see exactly what happened. And so that one, it's not, the cells that we inject kind of go away after a while, 2, 4, 6 weeks. Depends on the model. I've shown them up to 6 months, but they go away. But there's an effect that lasts, and I think what we're doing is we're transforming what's going on here at the cellular level of these resident macrophages that are in the heart. So just to tell you, the way we put these cells in is we map the heart with this 3-dimensional real-time thing that generates a map like this. And we go into the heart with a little catheter, has a little needle. You can see that, and this is a real map from somebody that got cells. The black dot, the dots are where we injected the cells. We can color code this with voltage, so you could see red is low and purple is high. This guy had a heart attack in this area, and we're kind of circling the injections around where the heart attack was. So we're kind of somewhat sophisticated in how we deliver and sort of develop the strategy of how to go about doing this. So this is the original DREAM trial that was published in the Journal of American College of Cardiology last year, okay? And I will just show you some highlights. So, you start with 1,100. This trial took 5 years to do. 1,100 patients. We have 565 that are randomized, but there was a month delay between randomization and actually treatment, so some people fell out. So our analysis population is 537 patients. So 265 were randomized to receive MPCs. MPCs are these mesenchymal precursor cells. 272 were control patients. And I just want to show you this because you're going to remember it when I show you some of the data, that ischemic etiology, remember we talked about the injury from ischemia, okay, and non-ischemic etiology. All right, so this is the breakdown of these patients. But basically we have 2 groups, and these are blinded. The patients don't know what they got. The doctors don't know what they got. We got a group of people doing the cells. We got a group of people following the patients. So this is done to the highest rigor possible. It wouldn't be published in this journal if it weren't. So this is the kind of thing that we used to get. It's called a surrogate endpoint. This is how well your heart pumps. If you just look up, this column here has all the patients. And this is the bottom line right here, ejection fraction. And what do we see? We see that the guys that got the cells pump significantly. So a P of less than 0.05 is significant. So there's a significant improvement in the pumping function. And we've been doing that for a while, and everybody goes, yeah, but we need to see more than that. And these 2 columns, so HSCRP is a marker of inflammation. Okay, and we know, because I showed you that complicated side with all that stuff going on, that's basically inflammation and immunity. We talked about adaptive and innate immunity. That generates inflammation. That's what starts heart failure. Is what keeps heart failure going. And so when we separate the patients out in guys that have more inflammation and have less inflammation, then the benefit is really pronounced in the people with more inflammation. So that's something to keep in mind. Okay, so let's just just to kind of confirm this, and this is coming out in a paper that's accepted in the European Heart Journal that's just submitted. It's not in press yet. So what we did is we took some further looks at this, and I'll show you why. But we wanted to look at the control patients. So these are people that didn't get treated, and we just want to see the effect of inflammation in those people. So talk to you about hsCRP. Okay, this is death. Okay, so these are patients that had both CRP and IL-6. It turns out that IL-6 is a pro-inflammatory cytokine. It's a bad guy. It's a bad chemical that incites inflammation. And it actually, in the liver, it turns into CRP. But we needed to make sure that we're measuring the same thing. What if I measure this and one was low and the other was high? Uh-oh, I'm in trouble. This is just nonsense, right? But it's not. It's identical. And so what we see here is, wow, in the guys that have high CRP, look at the mortality compared to the guys with a low CRP. IL-6, the inflammatory marker, high mortality, much lower in the guys without inflammation. So this kind of confirms our thoughts about inflammation and mortality. Forget treatment. This is just over 5 years what's happening with these patients. Okay, so this is the graphic representation of what I just showed you. These are controlled. Nobody got treated here. Okay, this is 5 years. This is the guys with low CRP, and these are the guys with high CRP. So this is mortality, and these people are dying a lot more, significantly more. You can see the p-value right there, than the guys with low inflammation. So this is important for us to really get to the mechanism. And as an aside, what's interesting is if you take this low curve here of the CRP, and now we put the guys that had CRP that got the cells, look, they're the same. So we've changed the guys that had high inflammation to guys that had low inflammation by giving them these stem cells. And mortality, okay? Now, that's not significant, okay? So that's a point that I want to make. So we're after this, right? So these are these two slides are the main results that showed it, but this was pretty impressive because we had never seen anything like this. And this is a lot of patients. So this is the whole 537 patients. Controls, and now we're talking controls versus treated, okay? 58% reduction, very significant in heart attacks and strokes. Wow. Okay, I almost fell off my chair when I saw this. Okay, so then let's divide it. The inflammation guys, wow, 75% risk reduction versus the non-inflammation guys, not significant. They kind of come together. Now let's add mortality to this. So now this is heart attack, stroke, and death. And you can see in the entire population, this is not significant. There is a 27% reduction, so it's going in the right direction. And if you look at the inflammation, it's almost significant, but it's not. Okay? So we're thinking, okay, now how, let's figure this out some more. So we looked, remember the mechanism and ischemia and injury and all that? So we separated out, showed you in that flow diagram, the ischemics versus the non-ischemics. We separated out the patients that had ischemia, okay? And if you just look at ischemia, and you can see control and MPC, the patients that had ischemia did much better, lower mortality, okay? Actually, not lower mortality. This is MI and stroke. And then when we add that triple endpoint, MI and stroke and death, it's significant now, okay? So looking at ischemic patients, we see a signal that ischemia, separate from inflammation, if they have blockage disease and those heart attack patients, they do better. And these are the non-ischemic patients. There's basically no difference down here. So now let's put the two together. Now we're starting to figure stuff out. We need to find out who to treat. So, heart attack and stroke, 88% reduction in patients with ischemic heart failure and inflammation. Okay? Heart attack, stroke, and death, 52% reduction in the patients that had ischemia inflammation. So we've kind of gotten, so we go to the FDA now and said, okay, we've figured this out. It says, okay, you do a trial with these endpoints, we'll approve it. That's Dream 2, which is in white. Because I had to do a new catheter system. They happened to pull out the catheter system, J&J, who's evil. They pulled out in an inexplicable way because they weren't making profit on this catheter. They took it out. So we developed 3 different. So I've been working for the last year, just kind of like, okay, stop everything. Let's develop a new catheter system to deliver the cells. We have it now. And so now we can start the next trial. So in summary, I'll summarize this in these slides here, in one slide actually. So what you see here is, this is heart attack and stroke. And so if we look at all the patients, okay, we see that with the cells, with the treatment, we get a significant event. We look in the ischemic patients, it's significant. We look at inflammation, it's more significant. But if we add the two, boy, we're really knocking it out of the park here. And if we go to the triple endpoint that includes death, again, same thing. All patients, not significant. Ischemia, significant. Inflammation, barely. We put the two together. Now we're talking. So we know, given, this is kind of like uniting the mechanism of action and how the cells work to what we're getting. Now, to the point about this talk, part of the thing is regenerative medicine, right? Okay, this is not regenerative medicine. This is anti-inflammatory and immunomodulatory medicine. We're doing other things, and there's a lot of exciting. So I don't want to give you the impression that this is the thing that's going to get approved first, okay? And it's nice to be able to offer something to a patient and say, hey, over 5 years, you got an 88% chance of not having a stroke, a heart attack, or dying. So that's probably pretty good. And you got to think that this is on top of all the medicine. We have really good medicines now. We're much better. This is on top of what we call guideline-directed medical therapy, which is all the medicines that we give people. So this is additive because we're addressing not the volume overload and people are got lots of fluid and they can't breathe and their lungs got, we're addressing the mechanisms in the heart. We're going into the heart, taking a fire extinguisher and putting out that inflammation, and the heart starts pumping better. And then we see effects systemically. You don't not have a stroke because your heart's pumping better. We're also affecting inflammation and blockages around the body. This is actually an anti-atherosclerotic therapy as well. That's a topic for a different day. Okay, so hopefully that gives you a broad idea of where we're going. And this is practical. Okay, this is going to happen. We're 25 years into it. But those guys that started running on the beach, we're going to get to an approval for these cells in heart failure, and that'll be the first step. And everybody in the world is made of 2 stem cells. Okay, so we know that cell therapy, if you can build a person, we can fix some tissue. Okay, so I know it works. So the real work here is trying to figure all this stuff out. And we're getting there slowly. So thank you very much for listening.
Thank you, Dr. Perin. I want to know where those 2 guys running on the beach, where they are now. Are they still running on the beach? So our next speaker is Dr. Kirstin Matthews. She's a fellow in Science and Technology Policy here at Rice University's Baker School for Public Policy. and a lecturer in the Department of Biosciences at Rice University. She is also a track advisor for the Weiss School of Natural Sciences, Professional Science Master in Biosciences and Health Policy, a core member of Rice Synthetic Biology Institute, and a steering committee member for Rice's Medical Humanities Research Institute. Dr. Matthews is the director of the Baker Institute's Science and Technology Policy Program and the Center for Health and Biosciences Biomedical Research Program. Her research focuses on ethical and policy issues at the intersection between traditional biomedical research and public policy. Specifically, she focuses on regulation and ethical issues associated with emerging biotechnology, including vaccines, stem cells, synthetic biology, and genomic medicine. Matthews also collaborates with Kenneth Evans and Neil Lane to understand how scientific advice is used and is provided for the federal government, including the White House Office of Science and Technology Policy and the President's Council of Advisors on Science and Technology. Dr. Matthews has a BA in biochemistry from the University of Texas at Austin and a Ph.D. in molecular biology from the University of Texas Health Science Center at Houston. Dr. Matthews, our series co-founder, will present the final presentation entitled Ethical and Policy Challenges, Reflections on the Past and a Review of Current Issues. Dr. Matthews.
It's a little intimidating to go after my last two presenters here. I feel like I'm a little off why I'm here, but what I want to do is frame a little bit about what's been going on in policy and ethics in the past 25 years within this space. A lot has happened, as the two speakers have mentioned, within science. We've had a great amount of progress. I think a lot of people started hearing about stem cells way back in the late 90s when embryonic stem cells were first isolated by a group in the US in Wisconsin. But we've seen great advances within this space over the 25 years, including the induced pluripotent stem cells that we've discussed. I guess it's supposed to be directed reprogramming, which I refer to as transdifferentiation here. Ways of organizing the cells, which we refer to as organoids. But there's a lot of prospect, I think, and Dr. Perin pointed out as well, both of our speakers did. It's really looking forward to this future that we're going to see in regenerative medicine and stem cell therapies. A lot more avenues, I think, than we would have predicted in 1998. I wanted to talk and I kind of frame this conversation in two different areas. I'm going to separate conversations about pluripotent stem cells, so those are the embryonic ones, and those pluripotent stem cells, versus what I would refer to as an adult stem cell, so the mesenchymal cells that Dr. Perin talked about. Those are ones that are more advanced as far as the cell's lifespan, what they can actually do, or what we've known they would traditionally do. And I'll start with the pluripotent first. We actually did have some resolution, I would say, in the past 25 years related to a few issues in this space. The U.S. federal policy first started with a lot of confusion at 1998, but then it started solidifying under George W. Bush, where he said that we could do embryonic stem cell research, but we couldn't create lines. And he had a certain set of lines. He said these are the ones you could use. When Obama became president, he kind of expanded the number that could actually qualify, but put over on top of it a set of guidelines. He says, here are the ethical guidelines that you have to follow in order for your line to be qualified to do research that's federally funded. And in that process, we actually really did a great job of solidifying what consent would be like in this space, what people should know, the fact that no one should be coerced into donating for this research, that you shouldn't, for instance, get free IVF in order to donate for an embryo for research, making sure that you donated and you knew that these cells would be immortal, they might even be sequenced, etc. Those lines that were focused, like, I only want this to go for diabetes research, the NIH said, okay, then they can only be used for diabetes research. You can't use it for general fundamental research. It needs to be more specific. In contrast, there's some new but also some unresolved older issues within this space. I'll talk about clinical utility a little bit next, research on the embryo itself, I think, and then this particular space that's really emerging and new called embryoids. So going with the clinical trials, as we've been talking about some of the research that's going on, there is a lot of clinical work, but it's a question of whether or not these will be the source that we end up with eventually in a clinical therapy. And this is just a quick snapshot of 1 year or 2 year space where there were 34 clinical trials. So there is research going on using pluripotent stem cells. This points out supposed to say embryonic, but a third of them are using embryonic and 2/3 are using the induced pluripotent. This study also helped us look at a decade of clinical trial research. And as you can see, there's about 109 clinical trials that were active in this time point. I can't tell you whether or not all of them are finished, but I do tell you that none of them have gotten approved for therapy yet. So some might be ongoing, some might have stopped. It could be like Dr. Perin's, where he showed us iterations of different trials. The most popular site, as you can see, is the top one, is the eye. The most popular actual specific disease is acute macular degeneration. I think the eye was extremely appealing because you have an automatic control. So they will do a procedure on one eye and leave the other eye alone. And then you'll be able to see if there's any issues within it. The site is pretty easy to find and locate. You don't have to do as much of the echo work that we saw Dr. Perin did. And so there's a lot of work in that space, and there's a lot of promise. But this has still been going on for over 10 years, and it still hasn't made it to the end. And you can see other in ischemic cardiopathy as one of the other ones in here. So it's really promising, but I will say that we haven't actually made it to that approved goal line. And as Dr. Perin pointed out, that's really, really challenging to make too. So that does actually make sense. The other unresolved issue really focuses in on the use of the embryo for research. I think just focusing in on the US, we still haven't really decided what we approve and do not approve of. There's lots of contradictory views and values within this space. But the discussion about embryo research came up again in 2016 when a lab was able to, actually 2 labs, were able to grow embryos to 14 days after fertilization. So this point is where the cells come together, but they're not actually fully embedded into the woman's womb yet. If you're talking in utero, they're able to grow, they're starting to specialize a little bit. And this has been actually a consensus deadline. We don't have it in law in the US because we actually don't fund any embryo research federally in the US. But it actually is embedded in several laws in different countries. For instance, the UK has a limit at 14 days as a law. So the question became, well, should we actually expand the research and allow for more research to go beyond this time point? Is the actual time point itself justified? There was a lot of work done to create that particular thought and philosophers and religious scholars. And I can speak about that if you have questions during the Q&A or later. But that's still kind of an open, unresolved one of where we should be. So the US still doesn't have a specific policy on embryo research beyond the fact we do not have federal funds for it. And then we also had researchers doing this really cool novel process, and one of them actually is based at Rice University, where they took these stem cells, these pluripotent cells, and were able to coax them into starting to develop like an embryo in different weird ways at different time points. They can partially replicate an embryo, some of them better than the others. So the one at the top is from a lab at Rice. It's 2-dimensional, and since we're not 2-dimensional, you can tell you that doesn't look very much like us, but it has the 3 cell layers that are present around 17 days. So he's able to study these kind of a black box time period where we don't know very much. We know a lot about what happens the first 5 days because that's the area of IVF. And then after that, it's kind of fuzzy about development until we actually start getting more information when an embryo is larger, or a fetus. And the second one at the bottom is actually mimicking a 5-day embryo. So it's actually more, you can see 3-dimensional. It's still missing some of the features. And I recently saw an article where they said it's like you see it, it looks like a house, but you walk inside and the walls are crooked and the floors are crooked and everything is kind of sort of off a little bit. So that's actually where we are at the moment. But the problem is that we don't know if it works really well until we grow it longer. And then when we grow it longer, we'll start feeling uncomfortable. They're doing longer term using animal models, but then the question is we have regulation for animal research. So if you use research like Dr. Srivastava mentioned with mice, you have to go in a protocol committee. So if you start with cell lines and then end up with something that is more of an organized, actually, mouse that looks like a mouse fetus, does that need to have some kind of animal subject's approval at that point? Like, when does it come in? Because the cell line's just, a couple things in a petri dish. So how we regulate it is going to actually be kind of more complex as we think forward in the future. When does it become something that we feel like closer to an embryo is another question. And then there's been a lot of discussion about how this will decrease the use of embryos. But if you're using embryos as a control, then in fact it might actually increase a little bit. But we're able to, the reason why they're also interesting is because they're able to actually grow large numbers of them in a plate at the same time. But I really feel like framing this whole thing, and again, especially the second part, it is revolved around our questions about when life begins. We have differing opinions, and if you actually go through monotheistic religions, there's differencing of opinions all across them as well. If you're a Christian conservative, you will say life begins at fertilization. At this point, we're starting to see life being created. And so all the research using embryos becomes really controversial. Other religions look at ensoulment happening at a later time point. For instance, like 40 days is where you are ensouled. So this isn't really as controversial, but yet when we start growing these embryoids or embryos in a dish to later time points, that becomes concerning as well. So how do we treat these as special, right? They are special because they are humans in some fashion because we're putting our genetics together. And these are going to be the unresolved issues, which I don't know if we'll ever really resolve, which frames a lot of these questions. And I'm worried in the nearer future about how that impacts a couple of different areas. In vitro fertilization used to, what I always thought was a safe space. Everyone wants to create babies. Everyone loves babies and wants to help people who are infertile have babies when they want to have them. But in the past year, we saw with the Alabama Supreme Court that they viewed life beginning at fertilization. Therefore, the IVF clinic is liable for when they got destroyed as actually destroying a potential human, a baby. In addition, we saw the Southern Baptist Convention this summer have a discussion about whether IVF was immoral, deciding it was. So these are going to be interesting times as we move forward. I think it's definitely going to be based on who is in control of the White House, the Congress, and even in Austin about how we are going to start looking at this particular space that I would say is probably underregulated just because we can't decide when life begins. So therefore, we have a hard time figuring out how to deal with that. And as we deal with that, the other questions about embryo research, and then again about fetal cell research, which is a little later time point, are also going to be coming under a microscope. So everyone then goes, well, adult stem cells should be fine. These are later time points. They don't involve the embryo. There shouldn't be any issues with these whatsoever. And unfortunately, there are a few, and it actually kind of links into some of the research that's going on. So this is, again, mesenchymal stem cells. I wanted to point out the number of trials we see here in comparison to embryonic is significantly higher. We saw about 100, where here we're seeing over 900 trials in this particular time period. But most of them, there's not a lot that are in the phase 3. Dr. Perin can tell me which ones are his, all of these years. And they're focused on neurological, cardio, and joint within. But we're not actually we're seeing publicly a lot of clinics who are promoting these actually for a lot other different therapies that don't have any kind of clinical trial approval. We're seeing celebrity endorsements. I like this one because it shows a Chinese woman turning into a blonde woman over to the side. I was just like, I don't understand how. We're seeing them in other countries, but we're also seeing them in the United States and Texas. Clinics that are promoting stem cells as just like a panacea. It's going to fix everything. It's going to make you live longer. It's going to help your complexion as an anti-aging technique. It's going to help a bunch of other areas. And then I think most disturbing is kind of a huge smorgasbord when you go through dozens and dozens of different diseases it feels like it's going to impact. And so you're thinking about, and I guess we'll ask the scientists, I don't know where autism fits within the space of using a cell regenerative therapy. I don't feel like we understand autism enough to be able to think that this was going to be done, but it's preying on parents whose child is having a hard time functioning within society, and here we can just give you an injection of embryonic stem cells or mesenchymal cells and cure it. And this is really the troubling space we have within here. These clinics are promoting stem cell-based interventions that haven't been tested like Dr. Perin does. That we can't guarantee they are safe, they work. They oftentimes don't have the same kind of medical expertise. You're going to a foreign country that doesn't have an FDA. You're going to a clinic that's run by a chiropractor to give you stem cells. Seems like a little odd, and they're injecting it into your heart. I think I'd rather have Dr. Perin do it than going to some random person. We have a lot of health effects have been identified, so these aren't just benign. Sometimes they are, sometimes they do nothing, but other times we actually have significant side effects, including deaths that have occurred from these particular treatments that are going unregulated. They're extremely expensive. Dr. Perin can tell you how pricey these things are, but when they do it in these smaller clinics, they can cost $10,000 per injection, asking you to come multiple times. They can cost up $100,000 if it's not in your city. If you had to travel to the location, then you have to add all that into it. And none of that's covered by private health insurance or Medicaid and Medicare, because this is experimental. So all of that is actually being covered by the individual. I would say the emotional toll because of the hope that's raised, but also the financial issues. And I do think it actually impacts research, because if, or maybe you went to China, or maybe you went to Malaysia and got a bunch of cells injected into you, I don't think Dr. Perin's going to be very excited about putting you in his clinical trial, because he doesn't know how those cells that we can't define impact what he's doing for you. We're trying to be able to do these as clean as possible, and the ability to understand what's happening with the particular therapy in a clinical trial. And by kind of playing around with something else, the lack of standardization means they can't even tell us how many cells are alive that they give an injection. So they just pull it from one source and put it into another source. States have been actually thinking about this policy since we don't have a federal one. We do like to do a lot in two ways that we're approaching it. So one way is creating labels. So telling people who go to clinics that they are actually going to some place and getting an intervention that hasn't gotten approved by the FDA. So we see legislation in California, Washington, Vermont, and Utah that does that. And then Vermont and Utah also will have advertisements with a label on it saying this wasn't approved by the FDA. But there are questions about the impact. If it's on a clinic, yeah, by the time you walk into a clinic, you made an appointment, you got in, you've decided to do it, it's probably not likely. You may or may not have actually read all the signs on the wall in the clinic, for instance. In addition, it's a question of whether or not these particular patients actually trust the FDA system in the process, right? Is that the best broker of this information? Or would it come better from, for instance, Dr. Perin as a spokesperson saying that this isn't going to work? Unfortunately, some other states do the opposite, which includes the state of Texas, as actually expanding access to experimental treatments. Utah, as you see, is in both groups, so they do have like labels on it, but while allowing it at the same time. But Texas does not yet have, as well as the other states, anything, so it's hard to tell whether the patients really know it's unproven. We did have, there's a great article in the Texas Tribune, or no, it's a Texas Monthly, about some patients who received stem cells and got huge infections and didn't realize that that's what they were getting, an experimental treatment, when in actuality it was. So I'm going to try to be shorter than the other ones. I also want to note Dr. Srivastava did point out the idea of equity, as well as being able to get access to these treatments that are going to be extremely pricey. But while we've kind of had some consensus on a few policy issues, I think there's a lot out there that maybe are unresolvable because of the differences in society, but figuring out how we're going to approach them in the future, I think is going to be an interesting challenge. And I'll leave the rest for time for Q&A.
Thank you, Dr. Matthews. I'd like to invite our speakers up for our question and answer portion. Wow, that was fascinating. And I guess lots of questions. I'm sure that you all have quite a few questions yourself. I do want to start with an initial question for both of you of what do you believe, looking forward, are the most exciting innovations that are upcoming this year and in the next few years? We can start with you, Dr. Srivastava.
Well, as I mentioned, a lot of the things that have been developing through the laboratory work and in animal models for the last couple of decades, really, are now in clinical trials. And so I think, you never know until you're in clinical trials like you've seen how that'll transpire. So I'm very excited to see how those read out in the coming years. And like Dr. Perin mentioned, when you do the clinical trials, not everybody is the same. Every one of us is so different. And the origins of our diseases are different. And so being able to properly stratify the populations that are being treated so you can do the analyses to know who does it really work on and who doesn't work on, I think is going to be key. And so to beginning to also genetically stratify those people, now that we can know everybody's genomes and their sequences, I think that'll affect what they're bringing to the table. And so I think the combination of knowing genetics, knowing deeply the mechanism by which you're making potentially making an improvement, and combining those, I think will give us a lot more power in the trials.
Well, yes, the innovation also of genetics and knowing those genomes, you can now swab someone's cheek and figure out which medication is helpful and which one isn't, Tylenol or Advil. I mean, it's really happened in the past couple of years, right?
Just a few years, and it's because the cost of knowing your genome and genetic information is plummeted. The first human genome sequencing, where you decode the 3 billion pieces of your DNA that make up your genome, cost us $1 billion. Just 20 years ago, $1 billion. You can now go in and know your entire code for about $300. So that's changing everything.
Does insurance cover that?
It's really interesting. Not yet. So this is a case where the financial part of the industry has not kept up with the science, where if you go to your doctor, I won't think twice, you won't think twice about checking off a box of a test that costs $200-300 for a blood test, to look at your blood count. I won't even think for a millisecond about it. And we'll order 10 of them that might be $1,000. And insurance won't think about it for a second to pay for it. But today, even though, unlike a blood test where you do every year, you would need to do this once your whole life, right? Every baby born in the United States should have it done day one, right? $300. We spend thousands just leaving the hospital as a newborn, but insurance doesn't cover it right now. And doctors don't think about doing it.
So what do you think it's going to take?
It's going to take education, both on the physician side, but also on the insurer side, and providers to realize that if they knew, like you said, that from day one, that you're going to respond to this medicine but not to this, then you'd save those $300 in a millisecond. Yeah. So I think a lot of it's just education and realizing that the landscape's changed, and it just takes time in medicine, oftentimes. And Dr. Perin could speak to the time it took from where we knew lowering your cholesterol would be helpful to when doctors actually implemented, cardiologists actually implemented it. It took decades. So
decades. So let's talk about that.
So I'm excited about a lot of things, but in particular, I'll bring up one thing, and I talked about cell therapy, but we, at Texas Heart, this is a homegrown, there's been a lot of research. Dr. Jim Martin figured out the pathway in, when you're born, you've got all this signaling that happens, it's genetic, it's incredible, the body's amazing. And so when you're born, the number, your cells, you know, when you're in the embryo, you're really small, and then you're a baby, and so there's signals that tell the heart cells stop multiplying the number of cells, okay? That just happens, it's we all have this. He figured out a way, it's an inhibitory pathway, to uninhibit that and say, you could start multiplying again. Now you can imagine somebody's had a heart attack. That's regenerative medicine. Now we get more cells to grow. Okay, so 2 weeks ago, we've been working on this for 10 years. Mice, pigs, you name it. Done a million studies on this, safety, etc. So 2 weeks ago, we submitted an IND, which is an investigational new drug. It's something you submit to the FDA for them to let you do research. And before that, you have pre-IND meetings, so you kind of know you're going to get a yes after, you know, because you talk a lot. And so in 2 weeks, we should have an answer, and this would be the first trial in a human being in which we put, it's in a virus, but this is, you take all the bad stuff out of the virus, you put the genes that you want to be expressed. It's very effective. And so we'll be able to inject this virus with this genetic code that unlocks the ability of the heart to start multiplying cells. And the beauty of this is that it's kind of like a river flowing. The body tends not to want to have cancer. And so this is not something that you turn on and it's a disaster and it grows forever. It lasts for about 2 months and then it kind of dies down. So with any luck, we'll get an answer from the FDA. They have to answer in 30 days. Maybe before the end of the year, we'll start the first gene therapy trial in a human being and grow of true regenerative medicine and grow some heart muscle cells. And that's very exciting. The whole frontier of gene therapy and cell therapy, this is all going to come together, okay? Time flies. And we're starting to, 300 years from now, they'll look at this and go like, oh my God, these guys didn't know what they were doing. But, you know, we're starting to get some basic understanding of how to use these tools, how to manipulate what's going on, and be able to apply them to get a result. Because you know what? I go to clinic, people die. People have heart failure, they can't breathe. So these are problems that we need to do what we can with what we've got. And at least we're getting to a more intelligent, knowledgeable point where it's acceptable to the regulatory agencies because we sort of understand the mechanisms a little better and know what we're doing. So.
And if I could comment, it might be interesting for you all to know that one of the beauty of science and medicine is how people work together to achieve these difficult goals. So Dr. Martin, who Dr. Perin just referred to leading this trial, who figured out this way to get cells to divide, kind of like I mentioned, he and I actually trained together here at MD Anderson, the same laboratory 30 years ago. And we have worked together with the same approach, but using that the LNP that I mentioned that was red in the heart that you saw as a delivery vehicle for this same type of therapy where you might be able to just inject it in a vein and it goes into the heart and gets cells to divide right there. So we're doing those tests right now in animals, and if that works, then we'll give it to you to do it in people.
I think the great thing about all this is the fact that you guys are all approaching cardiovascular from all these different ways. It's very similar-ish. Probably from our audience could probably think it sounds really similar, but it's all those at the same time to see which ones are winners or multiple winners, because your therapy might work a little bit better using the virus, while Dr. Srivastava's version might work better on other individuals. And as we get further along, we'll be able to use that knowledge to figure out who, like, personalized yours versus a medication that's personalized. It's the therapies that are going to be personalized even more.
Well, and then hopefully you said 2 weeks, you'll get an answer from the FDA.
Yep.
All right, so next year you come back and we can discuss
We'll
it.
start the trial. Now, something like this, when you do something for the first time, You do it very carefully. So we do what we call a pilot study. So we're going to do a patient. We wait for a month. We send the data to the FDA. They look at it. They're happy. We do patient number 2. We send the data. So when you do wild and crazy stuff, well, it's not wild and crazy stuff, but this kind of, it's very slow, very we're really, we don't know what we're going to get. We've done as much as you can in animals, and we've had long-term pigs. We treat the pigs and they go out to College Station to pasture,
Or to A&M.
and then we have to wait and bring them back and make sure there wasn't any cancer somewhere and all. So we've done all of this work, but you really only know what's going to happen when you put it in people. That's a very, very important thing. So.
And I wanted to say is that what we see, and the reason why I think we saw some of that state laws that were pushing for expanded access was because the frustration with the FDA, and I'm sure it's not easy to work with, but you appreciate that you don't want to put a dozen people into this trial at the exact same time in case you go, oh, whoops, there was this one little thing, right? It's an iterative process intentionally because you're actually working with humans. We want to protect them. And that's why it takes so long. It's not that the FDA is sitting around. They're responsive within 30 days, digging through the data, making a decision for you. The trial itself takes time. The data collection, watching the patients, and that's why it takes a lot to get through to a therapy at the very end. It's not that there was a big pile of paperwork you gave the FDA and they sat on it for 7 years.
It's an iterative, long-term process. But I think the FDA wants to. They're collaborative. They're kind of like the goalkeeper, and so they, are trying to protect patients and safety, and that's their role. And we respect that, and we have to work with them with that. To your point about the clinics, so we're doing all of this for a new treatment, but you can go to some ridiculous Mickey Mouse clinic here in Texas, okay?
There's quite a few.
And the brochures, man, I wish our brochures were as good as it, because it's beautiful. And they list every disease that they treat. And like for one, it's for cell techs, you go to Cancun to get your cells infused. Now, that's the mecca of stem cell therapy. So, this is about one thing. It's about money. So people exploring other people's emotions, desperation to make a buck. And that is just terrible.
So I wanted to open it up to the floor to see, I knew people would have some good questions. All right, well, let's begin with you.
I want to thank all three of you guys for supporting a wonderful talk. It was very informative. I have a question for both Dr. Matthews and Dr. Srivastava as well. Dr. Matthews, you mentioned in one of your graphs that in recent years, there have been more trials using induced pluripotent stem cells rather than embryonic stem cells. Could you elaborate on the difference in attitude you see towards trials and research that use these induced cells versus embryonic cells? Is there a kind of kinder attitude, or is there more leniency? What is the attitude you see from the public and from policymakers on that?
I think it's twofold. There's a little bit of an ethics, right, that embeds within this. Some of the trials, they started with those cell lines that were embryonic, but there's also that personalization that you can get with IPS cells, induced pluripotent stem cells, that you won't get with embryonic. So I think that also makes it really appealing. So for instance, I mentioned the eye, Dr. Srivastava could talk about his. You can actually use their own cells, put them in a dish, create them, and then put them back, which means that the immune system's less likely to kick them out. And some of the work is done in places where they have limitations on embryonic stem cells as well. But I think it's a lot more that you can actually do a personalized. But Dr. Srivastava, you're working on IPS, so why did you choose that?
I think those are some key features. And another key feature is just a little bit less scientific, and that is that Shinya Yamanaka, who I mentioned at our place, who invented these induced pluripotent stem cells, is from Japan. Japan took this as a huge source of national pride, that one of their scientists made this discovery. And so there's been a concerted Japanese government-led push to get these into clinical trial. And so the part of it is just as simple as that there's just been a lot of investment and focus from the Japanese government to get these into clinical trials, and that's been a big part of those numbers.
Motivation.
Yeah.
Government motivation.
Yeah.
So policy and politics.
And leadership. Yeah. It's policy. It's policy. Yeah.
Yeah.
That's exactly what it is.
Well, one small point about the iPS cells, embryonic cells. So the mesenchymal cells I talked about, the beauty, they fly under the radar of immunity, okay? If you take an animal and you inject mesenchymal cells in the same species, mesenchymal cells, they're cool, nothing's happening. You put iPS cells or heart muscle cells or whatever, you get rejection. That's like transplants, rejection was a big problem. So in doing these IPS, you're doing a heart transplant a cell at a time, kind of. And so and this is something we struggle with. So we're setting up these protocols, and I'm talking to these companies, and they're like, well, I think we need to immunosuppress these patients, and that's what we're doing in the beginning. But we don't know how much, what to do, how to immunize. I mean, so it's a little bit more complicated because you're giving cells that the body is going to react to. And we don't, and I wish I could tell you exactly what the recipe is. We've got several, and we're working on it, yeah.
Yes.
It was my wholly subjective impression that a year ago Dr. Perin seemed considerably more frustrated with the approval process than he does now.
Is that true?
It's a journey.
I said it was my wholly subjective impression a year ago when I had the privilege of hearing Dr. Perin speak that he was more frustrated with the approval process than he seems now. I was wondering if he could expand on that. And thank all of you. This is a wonderful presentation.
Thank you. Yeah, well, we got more data, and we hadn't talked to the FDA. Well, I can tell you a lot of things. But for example, we put in for an accelerated approval so there's different pathways in the FDA, and you can say, listen, we need a rapid, can you fast track this thing for me? They said no. At the time when they said no, we're not going to do it. So then we came again, and we did. I mean, we're persistent guys. And so we got, and now we have more data. And now we've met with them, and we've given this 80-page thing about this trial. And actually, the head of CBER, who's the division of the FDA that looks at biologics, changed. And she's an immunologist. And all of a sudden, I'm dealing with people that kind of understood better what we're doing. And so there's been a bit of a shift. You know, they're always careful, but there's been a shift of understanding. And we now have sort of light at the end of the tunnel. We still have to do a confirm. You know, you do the 600 patient trial over 5 years, and you know you get the answers like, okay, that's great. Here's the endpoints that we want. Do another one. And so you know how much that costs. But that's okay. And I think we do need a confirmatory trial. Little details I didn't talk to you about. For example, that triple endpoint, that's an ad hoc endpoint. It wasn't pre-specified. So scientifically, that's not as strong. So now we have to go, when you do a clinical trial, a phase 3 trial, you have to guess and lay out in exquisite detail everything that you expect to find and what you're going to analyze. You can't do something and go like, oh, this didn't work, so forget about that. I'm going to look at this. No, you have to lay it out perfectly. So this is exactly now working with the agency. They've told us, okay, this is what we want. But I'm good because this is working. So we know now what the recipe is. We're going to do exactly what they need to do. And if we can just see what we saw before, we're there.
And hopefully there's no transition in who is the decision maker at the FDA, because that makes a tremendous difference of that understanding that you mentioned.
Hopefully there's some consistency over time, you know, and so
But the data will sell itself.
But the data is the data.
Yeah.
You show them pre-specified mortality. They want a triple endpoint: heart attack, stroke, and mortality. If we can show that triple endpoint, completely pre-specified, done in a very blinded, very rigorous way, and we reproduce that and show that, they'll approve it.
All of you at some point mentioned the financial implications, and you specifically called out a particular company with J&J and the issues of the profitability. How is this being funded, and how is this being paid for?
everywhere,
Anybody,
public, private, probably some even individual donors perhaps, all the way through the process. Dr. Srivastava is probably predominantly off of the public, NIH, National Institutes of Health, funds the basic research predominantly in the US, and biomedical, which they probably also help with some of yours. You also have to have companies that work because these expensive trials. Private donors. He's Roddenberry. You were talking about science fiction. He got funded by the guy from
That's
Star
cool.
Trek.
I know.
Trek
I'm
fan.
jealous. I want a Star Trek donor.
So tell us about Roddenberry, because some may not know.
Yeah, so Gene Roddenberry, who's the creator of Star Trek, after he and his wife passed their son Rod Roddenberry, created a foundation to honor his father, and their first big gift was to support our stem cell center some years ago, because they thought that this field, stem cell field, really reflected Gene Roddenberry's vision of science helping humanity in some futuristic fashion, and a lot of that's coming to reality now.
It's very generous and gracious.
They have.
Yeah. We've had some funding from different places. So 12 years we're part of an NIH network, so that's government. We have a trial ongoing with the California's a little bit more liberal. The CIRM, which is the California Institute of Regenerative Medicine, they have funds for these kind of things. And we're doing a trial with the people in San Diego using matrix, which is like the juice around the cells, to put in legs to get people to walk better. Anyway, so you've got government fundings, and you have philanthropy, which is something we're all after. Texas Heart Institute, we live on philanthropy. And then I always say, Apple invented the iPhone, not the government. And so industry, I love working with industry, okay? And so industry is a big, they're gonna, we gotta keep kind of a firewall, because it's like, okay, I can't, if I'm making like a gazillion dollars if this thing works, okay, now I'm totally biased and you can't believe what I say. But if we keep a firewall, and so like Mesoblast, the company that has these mesenchymal cells, if they make a gazillion dollars when it's approved, I get to present the data, and I'm very happy doing that. That's my profession, and but I don't get to make the millions of dollars. So funding is very tricky, and we get it everywhere we can.
I think on the same note, in the long term, all of you have mentioned that these therapies need to be financially accessible for patients themselves, and this is an issue also with CAR-T and all these cancer immunotherapies. So to make personalized medicine accessible in the long run, do you think it's going to have to be on the industry innovation side, or federal regulations, state regulations? How do you think it can become more accessible?
I would say I think these are going to be extremely expensive. I'm just going to say $1 million, something like that, perhaps. Maybe not your one, but the ones where we're talking about actual directed and regenerative, like the cell therapy version of it, I think might actually be quite a bit more than we anticipate. Sometimes the cost ends up being a calculation of how much life it would cost, how much life you have left, and how much would it cost to do other therapies and extending it. And that's part of these weird metrics that some of the pharmaceutical companies are using for it. And I think we're going to have to approach it at different ways. We've got quite expensive $1 million therapies out right now, and trying to decide, who gets access to them is going to be really complicated. Deciding if we need to regulate the pricing is going to be one way. Another way is, I think Dr. Srivastava can speak about this, is do we also need to rethink how funding works differently, and how we pay it, maybe how private insurance approaches these particular issues. Do you want to expand on
I'd say, growing up in Texas, even though I live in California now, I'm much more lean towards, not having regulations necessarily drive prices, but creating the right market forces. And so I think there are two ways I think people are thinking about this right now. One is if a treatment is very expensive, but you're going to live for another 20, 30 years, maybe you amortize payments. A company gets payments in a pay-for-performance manner, and if it stops working, they stop getting payments. So that, I think, is one way. The other way is to really say, okay, if I'm going to spend $1 million on this treatment, but over the lifetime of this patient, society is going to save $5 million, right? Because that patient would have cost, treatments for ongoing for that much, and maybe they're not able to work and be a productive member of society because of their disease. That's also lost to society. So you add that up and you say, okay, we're going to save $4 million. It's going to be a net benefit, but it's over 40 or 50 years. Can we somehow take a long-term view and say, have people invest, investors who want to make a profit, have them invest in getting some of that savings or benefit? So there's a profit, right? Society has a profit of $4 million. Can we say, okay, you take that risk and invest in it, and sort of like mortgage-backed securities that we have, people take 30 or 40 views with home mortgages, they get packaged and sold to investors who are going to bet on the profit they're going to make over that over 40 years, 30, 40 years. Innovation like that with financial instruments around healthcare could serve as a way to pay for this over the long term, and people take part of the profit. That's fine. Why not? And it gets paid for that way. So I have a lot of friends in that industry, because actually when I was at Rice, I was an economics major for a long time.
You can tell.
Before I decided to go to medical school. And so I'm getting them actively thinking about this. But I think that's a potential approach, but it'll take some innovation. And carbon credits is one sort of paradigm that the world has created, these carbon credits that now get traded. And there's a market for it, and the market sets the price. And so something like that, I think merging those two industries could be a solution. But it's early days, and innovation has to happen there. We will figure out the part to do this. But I'm imploring my colleagues in other fields to be creative, just as creative as we are in science, to solve this.
So it's cost-benefit analysis and also internal rate of return with return on investment and all of those things that you learn across the way.
Exactly. Yeah.
at the business school.
You went to the business school, so that's what. There's no reason we shouldn't be able to apply those similar conceptual frameworks to this.
To every industry, to propel it forward.
Yeah.
So I think we have time for one more question, and go ahead. I've neglected this side of the room, so go ahead.
Thank you so much for your presentation. It's actually my first time learning about stem cell therapy, and I really thought this was very concise and open to different perspectives from non-STEM background. But I wanted to learn more about the process of doing research. For example, I have two questions. Contamination in the petri dish with stem cells, like how is that approach, or when doing research with animals, mice, for example, is there a targeted audience, like age differences? Is there like pregnant animals, numbers of quantities? Like what is the targeted audience when researching?
So for the first part, contamination is really important, but we have ways in sterile settings to avoid that, and sometimes things get contaminated and we throw them away. but by and large, we can solve that. For the animals, we try to do, I think age is an important feature, and I think experiments are done on different ages of mice. Sex is an important feature, and we try to make sure we're doing experiments both on male and female animals, because they do respond differently, and clinical trials now are mandated to involve both males and females, because there was a period before that where all trials were only done on men. And we now know that women, particularly in the heart field, react very differently. Crash test dummies as well.
Crash test dummies were just on a male dummy. This is similar.
Yeah, maybe there's a reason for male dummies.
I didn't say that, you did.
But those are all really important features, and one's important to track.
I'll say that at Texas Heart, we do a lot of large animal, we like to call translational research, and so because you do the mice stuff. A lot of things work in mice, but the FDA wants to see a dress rehearsal, and so pigs we can use the same equipment that we're going to use in people, but it doesn't guarantee it's going to work. But it's kind of a dress rehearsal. Well, we have an animal lab, and actually I just installed $1 million plus new catheterization lab in the animal lab. Okay, I mean the sophistication of doing these large animal studies, because they have to be perfect, sterile. I don't even know if sometimes I'm looking at screens and injecting cells and looking at maps. I forget if I'm doing a human or an animal. Seriously, that's the rigor, the rigor of doing animals.
So your human patient
But look, it's
As long as it works.
The point is, it's the same equipment, right? So it's identical. So the rigor in which we do these animal studies is, I even joke with the guys in the animal lab that they're more rigorous about every little thing than they are in the cath lab. So things are done very, very, very carefully.
Question. I think I've read research from, I think you looked at Texas Heart Institute, talked to Paul's community. I think he worked with, may he rest in peace, with frogs. Like, is there STEM research also with frogs?
He might have. Usually in the animal lab, we do larger animals, so rabbits. We shy away from dogs. There was a time where we did purpose-bred hounds, but everybody loves dogs, and we don't want to, But dog hearts are unbelievable, man. It's hard to get a heart attack in a dog heart. You have to do some special things. But really mostly pigs, and then we use a lot of sheep. So like we just launched the first completely implantable artificial heart. This was news about a month ago. And that was all developed. I mean, they have a pump team. We can put a sheep on an artificial pump while we do a transplant or take the heart out and put an artificial heart. And then we have an ICU where that sheep is there for 6 months being monitored and cared for. So it's different animals for different things, but it's just a whole world. And needless to say, this is very expensive to do. Yeah.
The fully artificial heart, that was incredible news just a few years ago from the Texas Heart Institute. I'm sure you probably know. Well, I'm hopeful that everybody knows, because it was really kind of going from science fiction to nonfiction, where a fully artificial heart was implanted into a person that was waiting for a heart transplant, and it was very successful. So it's the innovation is spectacular in the rate that it's being developed.
Yeah, we have a little internal competition. See, they've got 2 big ORs down there. So one is the surgeons with their artificial heart stuff, and I'm on the biological side. It's like, oh, we don't need the metal thing. I could do this with cells and biology anyway. So it's a very friendly environment.
Well, thank you. I just wanted to thank the Baker Institute of Public Policy, along with the collaboration with the Texas Heart Institute and our incredible trailblazers, Dr. Matthews, Dr. Perin, Dr. Srivastava, and also Dr. Ho. We really appreciate you coming this morning for this really phenomenal conversation. I wanted to mention that this is being recorded and will be on the THI and the Baker Institute's website. So if you could please share it with your networks. I'd also like to take a moment to thank a co-collaborator on this series, Harry Sprung from the Texas Heart Institute, who is a dear friend of mine and fellow Rice MBA grad, so go Owls. And we look forward to seeing all of you next year. Thank you so much for coming.